Welcome to this in-depth explainer. Today, we're tackling one of the most absolutely mind-blowing biological phenomenons of our time, the science of young blood, or as it's known in the lab, heterochronic parabiosis. But we aren't just going to sit here and look at the theory. No, we are diving deep into the clinical translation blueprint. We're asking the really hard question. How do we actually take the raw, chaotic biology of cellular regeneration and engineer it into a precise, actionable clinical reality? Well, let's dive right in and figure it out. Here is our roadmap. We are going to move step by step down the clinical funnel today. We'll start by decoding the raw biology, isolate the signals responsible for tissue repair, look at 6 core clinical targets, navigate the harsh realities of biomanufacturing, and finally, see how AI is shaping the future of all this. All right, Section 1, Decoding Regeneration Biology. Let's start by wrapping our heads around the sheer scale of this biological puzzle. Imagine looking at systemic regeneration and initially staring at thousands of theoretical proteins. In the early research models of heterochronic parabiosis, scientists literally identified over 2,900 of them. But here is the harsh reality check from the lab. Tracking over 1,500 wildly fluctuating proteins is a mathematical and clinical impossibility. There is just no way you can build a reliable treatment out of that much noise. So the entire R&D journey really becomes about distilling that chaotic biological web down to a scalable lab-to-bedside commercial protocol. Moving right along to Section 2, Key Signals and Tissue Repair. To make sense of all this, we really have to zoom in. We have to pivot completely away from this popular myth of whole plasma therapies. The core breakthrough in our understanding is this: the regenerative effect that actually reverses aging in different tissues doesn't come from just giving someone generic whole plasma. It comes from highly specific biological triggers. We're talking about extracellular signals that cross tissue barriers to kick off highly targeted repair cascades in your bones, your heart, and your kidneys. Let's look at how this plays out in bone formation. Think of your bone health like a giant seesaw. On one side, you've got osteoclasts building bone up. On the other, osteoclasts breaking it down. Specific factors like GDF11, also known as BMP11, and BMP2/4 act directly on this seesaw. They trigger a pathway that directly promotes those bone-building cells. But crucially, at the exact same time, they suppress the signals that break bone down. So you are actively building new bone and actively stopping the breakdown simultaneously. It's a real game changer for increasing bone density and accelerating fracture repair. Now, repairing cartilage or vascular tissue. That also requires 2 things happening at once. First, you need growth activation, like IGF-1 triggering cell proliferation for cartilage. But second, and honestly just as importantly, these specific blood factors actively block the release of inflammatory cytokines, and they suppress the markers that cause cell cycle arrest. Think of it like a roadblock. You literally have to dismantle these senescence barriers before the cells can even think about restarting their growth cycles. And when we look at renal or kidney repair, it all comes down to energy. Kidney tubule cells need massive energy reserves just to regenerate. So the fascinating part is how GDF11 acts like the intake valve for a biological engine. It throttles up the master regulator for mitochondrial biogenesis. By sparking a massive surge in ATP production, those kidney cells finally get the raw energy they desperately need to restore function and inhibit fibrosis. So the science is absolutely incredible, right? We know the pathways. But this brings us to the most pragmatic, sometimes frustrating question that scientists face. How on earth do we actually translate these insanely complex pathways into something a lab can produce reliably at scale? Which leads us perfectly into Section 3, 6 core clinical targets. This is where the blueprint gets really actionable. Remember those 2,900 plus proteins we talked about earlier? To build a viable business model, we have to ruthlessly filter out the noise. We drop down to 1,500, then 212, then 50. And ultimately, commercial viability demands that we isolate just 6 actionable clinical targets. We have to focus on the handful of measurable, synthesizable factors that have the highest impact across multiple tissue types. Because at the end of the day, if we can't measure it and we can't make it, it's clinically useless. And since every single patient is incredibly unique, a broad-spectrum, one-size-fits-all treatment is just incredibly inefficient. So the core strategy becomes a 3-step protocol. First, dilution. We target and reduce the harmful accumulating senescent factors already in the patient. Second, diagnose. We run precise diagnostics to find out exactly what's missing. And third, replace. We introduce only the specific, targeted factors that specific patient actually needs. And here they are, the regenerative target matrix. These 6 factors, GDF11, EGF, VEGF, IGF1, PF4, and ADMTS3. They represent the absolute perfect overlap between what we can synthesize in a lab and what provides a massive systemic impact. GDF11 drives that mitochondrial energy. EGF and VEGF drive vascular repair. PF4 helps with neural maintenance. This matrix right here, this is our clinical playbook. Okay, section 4, scaled manufacturing and QC. Having a playbook is great, but executing it, that's a whole different story. So how do we actually get these 6 factors? Well, we can't just rely on off-the-shelf synthetic factors. They just don't have the clinical viability. We have to master the conditioning of natural media pools. We essentially have 3 main ways to do this. You've got platelet lysate, which offers high bioavailability but is kind of limited in specialization. On the flip side, MSC and EPC conditioned media are highly tailorable. Endothelial progenitor cells, for example, are absolutely ideal if you're trying to target specific vascular markers. Now, this brings us to a really fascinating concept called the oxygen paradox. In a standard lab, room air sits at about 20% oxygen. But for your internal tissue cells, 20% oxygen is actually hyperoxygenation. It's totally unnatural to them. To get our lab cells to secrete the maximum amount of regenerative factors, we literally have to stress them out. We engineer a strictly controlled hypoxic environment, dropping the oxygen way down to between 1% and 5%. This mimics their natural physiological stress and forces them to just pump out our target growth factors into the media. But as any researcher will tell you, a beautiful theoretical mechanism on paper is completely useless without a rigid, zero-tolerance manufacturing environment to back it up. Making the jump from a flexible research lab to a clinical production line is honestly a nightmare of variables. We literally call this the quality control gauntlet. In the lab, you might have some wiggle room. In a clinical reality, endotoxin limits are set at a shockingly strict 0.5 EU. Mycoplasma testing isn't just a simple yes or no. Clinical production faces massive risks of false positives just from harmless DNA fragmentation during PCR testing. You need total atmospheric isolation. Because I kid you not, just opening a door at the wrong time can alter the entire protein output of your batch. Finally, section 5, AI and the precision future. This is where it all comes together for the patient. If we can survive that manufacturing gauntlet, we reach the ultimate conclusion of this blueprint. Because every patient's protein degradation pattern is completely unique. One person might be desperately low in GDF11, while another is missing VEGF. We can't rely on one universal young blood serum. Instead, we have to maintain a diverse database of distinct media profiles that can be prescribed based on the patient's exact diagnostic gap. And since every single cell culture produces a slightly different protein yield, we use machine learning to map it all out. The AI will instantly read a patient's specific blood profile, map it against our database of batches, and instantly select the absolutely optimal batch of conditioned media to perfectly fill their biological deficit. It's incredibly precise. So, this is the translation mandate. We started with a raw, chaotic noise of 2,900 factors. We distilled them down to 6 actionable targets. We engineered hypoxic manufacturing to actually produce them. We pushed them through the zero-tolerance QC gauntlet, and we arrived at precision prescriptive regeneration. This isn't just a scientific paper anymore. It is an actionable, scalable, regenerative model. So, as we wrap up this explainer, I really want to leave you with this thought. We are leaving behind the era of guessing with general plasma therapies, and we're entering a brand new era of engineered, AI-matched precision. When we can diagnose exactly which of these core factors your body has lost and replace them with zero-tolerance precision, how will that targeted replacement rewrite your own biological timeline? Thanks so much for joining me today, and as always, keep questioning the science.